So thank you very much to the organizers for having me here. I'm really excited to be here, and it's an honor really to be able to talk to all of you here about the kinds of research questions that excite me, and hopefully I'll be able to get some of you also excited about them. In particular, as you heard in the introduction, I am motivated by biomedical applications. And so I would like to, with this, invite all of you in to hopefully also be able to, solve some of these really critical questions that we have in the biomedical sciences right now. So in the biomedical sciences, we are right now in a huge data revolution. Where it is possible now through single experiments to profile millions of cells, where you have imaging data, where you look at images of single cells, proteins within cells, you go all the way to tissue slides, you go all the way to whole organs, and then all the way to maybe a whole body. You have all kinds of different modalities, like say in a heart, you can get an ECG, you can get an MRI, you can get an angiogram. So you have all kinds of different modalities. And just to also tell you a little bit about the scale of these data sets, there I have the graph of just the genomics platform at the Broad. So just one institute, just sequencing data, not even taking into account all of the imaging data. And you see there how it scales to hundreds of petabytes of data just from one institute coming out alone. And so we really need everyone's help to make use of all of this data. And it's a little bit different than in many other fields where maybe prediction questions are, your gold standard and that's what you want to excel at. In the biomedical sciences, it's really all about causality. And so here I just have one example of fibrosis. So fibrosis is a disease where the tissue stiffens. It underlies all kinds of diseases that you may know, and in fact is really the cause in the end of about 45% of all deaths in the industrialized world. And there are still no cures for fibrosis. Now, let's think about what it means to identify drugs for a particular disease like fibrosis. So as I mentioned, fibrosis means the tissue stiffens. And so what you need to do is identify genes or proteins that are the cause of tissue stiffening, and not genes or proteins that are changed through tissue stiffening. Right? So you want something that is the cause of tissue stiffening and not something that is associated with tissue stiffening but is downstream of tissue stiffening, because that would be a very, very bad drug target. So it's clear here that you really need to be able to understand what is a cause and what is an effect. And in fact, I just brought up here fibrosis, but really, this problem is underlying everything, any target identification for any disease. If you're thinking about cancer immunotherapy, we want to identify the perturbations or the interventions that move your immune cells to an active state instead of, having them go into this terminally exhausted state. If you think about rejuvenation, we want to identify which ones are the perturbations that move your cells from a senescent state back to a younger state. If you're thinking about tissue regeneration, you want to take skin cells, for example, and turn them into whichever cells that you need in your body, right? So it's always the same problem of having a particular cell in a particular state, be it the disease state, an old state, and you want to identify what are the perturbations that move you to a new state. Now, let's look at, our super powerful Gen AI models that we currently have. And I'm just taking a very, very simple problem. We all know that, good weather and warm weather is upstream of high ice cream consumption, and it's not the other way around, right? We all know that if I would take a cold and gloomy day in Toronto in the winter, and I would go outside and give out a whole lot of free ice cream, to increase ice cream consumption, that's not going to change the weather, right? Now, let's take here ChatGPT and try to ask it to generate an image of an ice cream stand where they're giving out ice cream for free, and you should all try it out. It will actually always put good weather in the background, okay? Because it doesn't distinguish, and they were not developed to distinguish between cause and effect. models, current AI models, are very good at conditioning. If you condition on high ice cream consumption or ice cream consumption, the probability of good weather goes up. But if I'm intervening on ice cream consumption, I'm giving out free ice cream, that doesn't change the probability or the distribution of weather, right? So really these models should be able to have, your standard distribution of if you actually ask ChatGPT to generate images of parks, you will sometimes see it with snow, sometimes with rain, sometimes sunny, right? But it doesn't do it if you're actually saying that there is also ice cream in the picture. And that's not a fault, that's just how these models, they were not developed in order to have to be able to distinguish between, what is a cause and what is effect and what happens when you're intervening on a particular variable. Well, that will not change something that is upstream of it, right? But this is, of course, critical. Understanding what is upstream and what is downstream is really critical. I need to be able to ask the model, what happens if I intervene on gene A? Is that going to change tissue stiffening? Is tissue stiffening upstream or downstream of gene A or protein A? Okay, so we really need new Gen AI models that can actually reason causally for any of the, or for many of the biomedical questions that we care about. So let me now set up the causal framework, the traditional causal framework, and how that relates to biological questions. So, the gold standard in biology, and if we really want to understand, say fibrosis, is we would like to build these gene regulatory networks. So any human has 20,000 genes. So we would have these networks with 20,000 nodes, and, this is context specific. So for a lung epithelial cell, this network will look different than for a skin epithelial cell, for example. It will look different in the disease setting as in the healthy setting. And we would love to be able to draw this full causal network, maybe even having differential equations on every one of these little edges, right? And then, really this we can understand it causally. So Sewell Wright developed the foundations of causal inference by, having a statistical model that, can describe the distributions you're getting out when you have such a directed network. And how you understand it, I have it there at the bottom, is, every node is a random variable, and its distribution is, the value of this random variable is given as some function of its direct parents. Okay, so if I have a gene 4, for example, it is some function of its direct parents, in this case, gene 2 and gene 3, and then some noise. Okay, so just a very simple model, and you can understand this model causally because it also tells you what happens if I intervene in the system. So for example, here gene 4 is all the way at the bottom. So if you intervene on gene 4, well then nothing changes upstream. Okay. However, if you intervene on gene 2, well, because gene 4 is downstream of it, it will actually change gene 4. Okay, so these causal structural equation models, not only do they describe the observational distribution, they also tell you everything that can happen when you're intervening in the system, right, just by propagating the effects. Now, the problem is, if we were to know this whole causal network on these 20,000 genes in fibrosis, well, then we could predict and understand which gene we should, or protein we should intervene on in order to change the system back to the normal state. The problem is we don't know the network. Okay? And so causal structure discovery is a whole field in itself, which tries to, from observations on the nodes, try to learn this underlying causal network. Now, I could give a whole talk on causal structure discovery, is something that we have spent many, many years on. But in particular, we've identified all kinds of negative results. Negative results that say things like, even in order to learn a causal graph from scratch, just on 10 to 100 nodes, you need something like 10 to the 6 to 10 to the 7 interventional samples. So think about, learning a network on 20,000 nodes. We simply cannot do it with the types of data sets we will ever be able to generate. Okay, so that approach somehow is just not the approach that will work out for these kinds of biological questions. And so therefore, my talk is not going to be about causal structure discovery. However, then we have to ask ourselves, well, what can we do? We still want to answer these types of questions. So maybe we don't need to learn the whole underlying causal network on these 20,000 nodes in order to be able to predict what happens if I intervene, if I apply a particular drug, if I intervene on a particular gene. And that's really what we need to understand, right? So underlying the real problems in, drug discovery or in any cell state engineering is maybe an easier problem, which I have here, which is the following, right? We would be happy if we were able to predict the effect of any unseen intervention, perturbation. So I'll use these two words exchangeably. Perturbation is the word that is used in biology, and intervention used in all kinds of other fields. So, if we had, and we would like to do this across contexts. So say if, you have in the fibrosis context in the lung, or in the healthy state, you have data where you have applied some drugs and some genetic perturbations. And you've done this in the mouse. It would be amazing if I could then predict what happens in the human. Okay, so that's what I would say as generalization to new contexts. Okay, I have many different perturbations, be it drugs, genetic perturbations, etc., that I have applied in one context, say in one cell type, or in one disease context, or in one individual, or in one, species in the mouse. And I would now like to predict, well, what happens if I apply the same drug in a human, or the same drug in a new cell type, or in a new disease context. That's one way that I would need to generalize. The other one is, the space of possible perturbations is huge. So it's believed that there are about 10 to the 26 many drug-like molecules. We'll never be able to apply all drug-like molecules in every context we care about. So we need to be able to apply some drug-like molecules and generalize from them to other perturbations that we have not seen yet. Similarly, for genetic perturbations, we have 20,000 genes, as I said, but we care about combinations of genetic perturbations. We'll never be able to do all combinations, two-way, three-way, four-way, five-way, of all 20,000 genes in every context we care about. So we need a way to be able to generalize to unseen genetic perturbations. Okay, so there are two ways that we need to generalize, so new contexts and new perturbations. And I hope it's clear based on the example I gave with the ice cream and ice cream consumption and good weather. These are not your standard generalization problems, right? I can only succeed in these if you can figure out what is upstream and what is downstream. Now, if I want to put these problems into, Judea Pearl's causal hierarchy, Judea Pearl being one of the godfathers of causality, in particular of thinking about interventions, this problem that I just listed out, which really underlies all of drug discovery in biology, is actually at the Neanderthal level in Judea Pearl's hierarchy. That's why I'm saying it's maybe the easier problem. So he has this really nice hierarchy, and if you want to learn more about causality, I think the Book of Why is a very nice way of getting started, where you have all the way at the bottom, that's what current neural networks are really good at identifying associations, say between good weather and ice cream consumption. The next level up is intervention, is predicting the effect that, what will happen if I give out a whole lot of free ice cream. Okay, and then the next level up is what actually all of causality, or most of causality is, or causality research nowadays is about, is counterfactuals. What would have happened had I not given out free ice cream? That's a much harder problem. And in fact, for the drug discovery problem, maybe we don't even have to go there. Okay, because we will do experiments anyways. We want things that are actually experimentally verifiable. And so we're at the intervention level of predicting the effect of what will happen if I apply this particular drug-like molecule in this context. So we're really trying to solve that Neanderthal problem here of predicting the effect of an unseen intervention. But note, you will not be able to solve it with your standard association types of questions or types of tools. Okay, so if I go one back, now, still solving these types of problems, it will be impossible to do if every drug has a completely independent effect of every other drug, right? There needs to be some type of underlying low-dimensional structure in order to be able to hope that there could be any solution to it that we can find, right? And similarly, across different contexts, from mouse to human or from a skin cell to a heart cell, et cetera, there needs to be some underlying low-dimensional structure that we should be able to extract and use in order to be able to predict and generalize across contexts of perturbations. Otherwise, there is simply, no way we can do any of these tasks. Okay, so how do we think about this underlying low-dimensional structure, and why do we believe that there should be some underlying low-dimensional structure? So, as I said, we can, at the cell level, we can collect all kinds of data, be it imaging data, sequencing data, et cetera. Imaging data being actually some of the cheapest ones. Now, here I have, with many different drug perturbations, there are very large-scale data sets where imaging data has been taken when you're doing hundred thousands of different drug perturbations and hundred thousands of different genetic perturbations across different cell types. And you get these types of imaging data as I have there. Now, it's pretty clear right here that every pixel is certainly not a causal variable, right? Because pixel 1 in one image has nothing to do with pixel 1 in another image. So it's very clear there that, the causal variables still have to be learned. We don't know what actually the causal variables are. Is it maybe the size of the nucleus, or the shape of the cell, or where a particular protein is localized inside the cell? Right, so the causal features, so the nodes actually of your causal graph, they have to be learned. We cannot assume that we actually know them. And so there should be some lower dimensional representation of what these causal features actually are. Similarly, in fact, if we look at sequencing data, well, sequencing data is collected at the gene level. And so until now, we've always just thought of, let's just build this large network on 20,000 nodes. But if you look at interventional data, and you get sequencing data, so these vectors, 20,000 dimensional vectors, where I get to observe the value of every one of these 20,000 genes, in fact, you will see that many genes, they move together, okay? So really, you could think of them as one causal feature. And also, when you're doing different interventions on particular genes, you will see that they will have the same effect downstream. It's just that we don't know what are these modules, what are these features, so we have to learn them again. But just by looking at the data, it is clear that there is some lower dimensional representation, that we shouldn't be modeling things just at the level of genes or just at the level of pixels when we're having these observational data sets. Okay, and so really how we're thinking about it is, these types of models that I have here at the bottom is you have your x's, which are your observations, be it at the gene level or be it pixels in an image. But the causal structure is really not at that level. The causal structure is somewhere in a latent space. It's at the z level. That's where we want to learn these causal structures. That should be something lower dimensional. And then, of course, we have noise variables as well, because everything, is, of course, stochastic in these systems. Okay, so the causal structure, we want to learn it at the z level and not at the x level. So that's the model that we're thinking about. And, this is causal feature discovery, or also causal representation learning, which is nowadays a huge field. So you see here, it's definitely a representation learning problem, right? I need to learn the z's from the observed x's. But also it's causal because, these z's have to be causal variables, and I want to learn a causal structure among the z's. So there is this nice marriage that I will talk about between representation learning and causality that is actually going to be needed. Now, I also already brought up here and also talked about it at the beginning, we have the luxury not just of being able to do interventions in the biomedical field where I can literally go and apply any drug to any one of my cells, or I can apply any genetic perturbation nowadays using CRISPR to any of the cells. But in addition, we often collect not just one modality like images, but we also collect sequencing data in addition and all kinds of other modalities as well. Now, the one thing that is maybe different than other settings, like say if we're thinking about different modalities like images and image captions, so images and text, they're often collected in a paired way. At least you have some images and some captions that you can use to then generalize afterwards. Now, for many of these applications, getting an observation means killing the cell. So once I get that image of the cell, I cannot also get sequencing information from it. Okay, so it's always, basically always, unpaired data. Okay, so you have to decide for this particular cell, either I'm getting an image or I'm getting sequencing information. So it's a bit more difficult where you're never getting to see the paired data themselves. But still we can ask, there should be something that we can gain from having multimodal data, right? Where I can take a population of cells, I have, say, applied a drug to it, and by chance I can take out some for imaging and some for sequencing. Right? There should be something that I can gain from having different modalities together, even if they're not paired. And in particular, and now here I want to first give the intuition, there should be something to be gained when you have multimodal data if you care about causal features. And I want to give first the intuition, and we had the intuition a long time ago. It took us a long time until we were able to prove it, that it's actually true, that multimodality really helps you to get at causal features. And the thinking is the following. So what is hard about causality? It's that what is not hard is to find associations. Right? You always find a lot of associations, but the problem is we need to filter out all kinds of associations that don't come from causal relationships. Right? It's about throwing out most of the associations and then only keeping the ones that actually come from causal relations. That's the hard part. Now, an association is more likely to be causal if you see it in many different modalities, right? Because, something that is causal, it will be there because it's in the system, no matter whether you're looking at the system from the top or from the bottom or from the right or from the left, right? So this principle of invariance, so let me just filter out everything that I cannot see anymore if it's not there in modality 1 and modality 2 or modality 3. I'll just filter it out. For now, I'm not trying to find everything that is causal, but at least what I'm finding, I want it to be more likely to be causal. Okay, so I'll just throw out everything that is not there if I'm looking at it from many, many different viewpoints. Okay, so what I'm going to keep is everything that is shared between different modalities, shared between different views. So invariant to how you're looking at the system. Okay, and now, of course, we're getting to multimodal learning. So I'll talk first about multiple modalities being, imaging or sequencing. But of course, we can also think of multiple modalities being interventions. Modality 1 can be when I applied drug 1. Modality 2 can be when I applied drug 2, etc. Okay, and you should already think that, if I have different interventions as my modalities, that should somehow carry more information about the underlying causal system than if I just have observational data where modality 1 is imaging and then modality 2 is sequencing and modality 3 is maybe some proteins, etc. Right? Because when I'm doing interventions, I have the same causal system and then I have an intervention on the causal system, so I'm only locally changing things. Whereas if you have many different modalities, right, you somehow have to patch together. I'm all looking for whatever is invariant at the population level. So let me first do different modalities. And I'm sure many people here work on multimodal learning in many different applications. We've seen this, now for many years. But I think what comes out of, the thinking about getting to causal features is, and thinking about, well, what I want to keep is what is invariant to how I'm looking at it, is that we're actually, and maybe I already put this up, so, there are many different ways of getting a shared representation or integrating different modalities, right? And often, in fact, what people do is, any say, let's take simple autoencoder models, right? You get to some latent space. Often what people would do is concatenate features from different modalities. Say, I get a feature from a single cell image and I get a feature from sequencing, and I would concatenate them or I would add them together. But the intuition from the causal setting of, hey, I'm looking for what is invariant to how I'm looking at the system really suggests that we should actually be using just one latent space, okay? Namely a latent space that is invariant to how you're looking at the system. So we're going to use a contrastive or a discriminative loss, depending on if you have paired data or unpaired data. So, I'll show, or, one of the examples that we did, this may be easier to understand than the cellular ones if you're not in single cell biology, is say you have a heart, right? And you have individuals who got their heart ECG and heart MRI. Well, now we have paired data. That's a much easier setting, where I can, in the latent space, have, say, a contrastive loss, which says, well, these two modalities, ECG and MRI, came from the same individual. So I'll make sure that I have just one latent space. In the latent space, these two modalities are close together for the same individual. But in fact, the same also works if you have unpaired data, which is usually the case in single-cell biology. And now you have to use something, a discriminative loss, right? Which says, okay, I have a population of cells. By chance, I took out some for imaging. By chance, I took out some for sequencing. I know it's the same population of cells. So in my shared latent space, I should not be able to tell whether you're coming from sequencing land or imaging land. So I'll just add, another classifier in the latent space, which punishes me if you can tell whether you're coming from imaging land or sequencing land. So I'm going to match distributions in the latent space. Now, you'll tell me, if I have a Gaussian distribution in the latent space, there's no way for me to align the two distributions, right? So it's already clear that I will need assumptions, which are, of course, easily satisfiable in the biological setting, but, rotation symmetric distributions are not good, right? So it's good to have different clusters of cell types, et cetera, because I'm going to match distributions in the latent space. Okay, but just from the causal viewpoint, it suggests, what are the things that we should be doing in order to get our shared spaces, our shared latent spaces, and what are maybe things that we should not be doing. And so we always use, either a contrastive loss or a discriminative loss to get one shared latent space, okay, namely a shared latent space that is then invariant to how you're looking at the system. So that's number one. Now, I said it took us a long time to actually be able to show that this is really, that this actually leads to causal features. And, I don't want to go too much through the theorems, but, we now do have identifiability results. So if you have many different modalities, X1, X2, X3, they're unpaired. Then under certain assumptions, in particular non-Gaussianity, right? We all know we need it. But then there are also some assumptions that probably just have to do with the proof technique that we have, might not be really required. We're actually able to show that we really get identifiability. So from different modalities, you can identify the causal features. So you can identify how many latent causal features you have, and then also the joint distribution over these causal features. And maybe you see here already something more, and it's always something that we like, when actually theory tells us that we should be able to do more than what we thought we were able to do when we were doing the applications. So you see here that we actually have partially shared latent spaces. So we have these latent variables, Z1, Z2, Z3, that are the ones that are shared among all modalities. That's what we were looking before, when we said, well, we'll throw out everything that is not shared. So we would have thrown out Z5, Z6, and Z7, and Z4, because they're modality specific. They're not invariant to how you're looking at the system. But here theory tells us that we should also be able to identify these modality-specific causal variables. Okay, and so once we had this, although this didn't give rise to an algorithm, we were able to then, develop new algorithms that instead of one shared latent space, we now actually have partially overlapping shared latent spaces. Okay, so now we're going to decouple the information between two modalities, say imaging and sequencing, or heart ECG and heart MRI, right? It was probably already clear to you that, well, a heart MRI probably contains some information that isn't there in a heart ECG, and vice versa. And we should be able to identify what is it, not just look for what is shared between them. And so now we have algorithms that can do exactly that. We don't have any proofs on these algorithms, but the algorithms suggest, at least in applications, that we're able to deconvolve what is information that is only in a heart ECG that is not there, that you can simply not get out of a heart MRI, and vice versa. And, so we haven't applied it yet to heart ECG and MRI because, we mainly work on single cell types of questions. But here's something that we thought it would be shared. So we do a lot of things with imaging, very cheap imaging modality that all we look at is the DNA, so what carries your genetic information. You just fluorescently label the DNA. Those are known as DAPI images. They're super, super cheap. Now, in cancer, you might know that, in cancer cells, you will have breakpoints. So your DNA will actually be broken in different locations. And so we thought from a fluorescent label of the DNA, we should be able maybe to infer how many breakpoints there were in a cancer cell. Now, there is another stain for just looking at breakpoints. And in fact, we were able to see that if you care about, and many researchers do care about this, of identifying how many breakpoints there are in a particular cell, you actually cannot get this information out of just DAPI stain itself. It is in the information that is protein specific. So in this case, if this is what you care about, you have to do another label, which is in this case, this particular protein label. So it's really important for experimental design, right? To know what information can if you have a particular downstream task, right? What information can you infer from a cheap and easy modality like a heart ECG? And for what might you actually need a heart MRI? That will really depend on your underlying downstream applications. So this is all for different modalities, right? So if I go back, this is when X1, X2, and X3, one is, say, imaging, another one is sequencing, etc. All of them are unpaired. Now, I provided the intuition before, if every one of these modalities is an intervention, maybe we're able to do more. We should be, probably, right? Because the intervention should tell us something about the underlying causal network, or should carry more information about the underlying causal network than actually just having different modalities. And in fact, that's the case. And so I'll show you another theorem now and then applications of it. But note here, we have pretty strong assumptions, right? Not just the non-Gaussianity, but we had all these assumptions about linearity in the causal model, which of course is nothing that will be ever satisfied in true life. We had, these linear maps down to the different modalities. And, I don't have to go through the details, but in fact, if every one of your modalities is an intervention, all these assumptions are gone, okay? In a fully non-parametric setting, you now get identifiability of the full underlying causal model. Without any assumptions on the distribution, etc., you can actually now identify the full underlying causal model. And the types of things that I like the most, as I mentioned before, we didn't have an algorithm that actually has guarantees of identifiability. In this case here, the theorem goes on. It says this particular algorithm will actually identify the underlying causal variables and the causal graph. Okay, and so what does this algorithm do? So it's a generative model, first of all. So what comes in, and this will be also important for the final application. So what comes in is a cell, say an image of a cell, okay? And what also comes in is which perturbation you're performing. So I tell it I'm knocking out gene 1, or I'm applying this particular drug, but let's just say I'm knocking out gene 1. And it's trained in the way where you do have some data, right? Some interventional data where, I can train it to map to interventional data which I have seen, where I'm taking a particular cell, adding this particular intervention, and out should come a cell that looks like where I intervened on gene 1. Now, the real difficulty is that, as I said, I never have paired data, right? So if I'm taking an image of a cell before the intervention, I will not get to see that same cell after the intervention. So we have to do something at the distribution level, and so that's why it's a discrepancy-based VAE, right? So you have a batch of cells, control cells, a batch of cells which have the intervention, and I'm going to match them at the distributional level because I don't have a pairing of one cell versus the other cell. Okay, and so this particular model, as you see also there, what you have in the latent space, or in the latent part, it actually learns a graph as well. Okay, so it does learn in there a causal graph, and you have the guarantees that that causal graph among the Z's is really the correct underlying causal graph. Okay, and the interventions are going to be matched to the correct causal variables. Now, what are the types of applications, and I will end with this, that you can now do? Notice that at the beginning I started with these causal intervention prediction questions, right? So what I would like to do is put in a cell. So I have some data of, say, skin cells, and I have some data of these skin cells where I did particular genetic perturbations. Again, I'll never be able to do all genetic perturbations, and I'll not be able to apply it to, measure it in skin cells, heart cells, and in all kinds of different disease conditions. So I would like it to generalize to new contexts, so to new cells, and I would like it to generalize to new perturbations. But now these models are generative, right? So, once it's trained, where in comes some image of a cell, and you tell me which perturbation you're doing, well, it can output or generates the output of how that particular cell I'm predicting will look like under this particular perturbation. And so, of course, then you can put in new perturbations, combinations of perturbations. You can put in new cell types once you have something like this. And so, in fact, these models, they can now do things like generalizing to new perturbations. They can generalize to new cell types, disease contexts, etc. They can predict what happens when you're doing combinations of perturbations. You can now use them for designing the design of perturbations, right? I have a particular end state that I care about. I'm putting in a disease cell, but really I would like to find which perturbation will give me a healthy phenotype. Well, if you can predict the effect of any perturbation, then you can do the reverse question of saying, well, which perturbation should I have applied in order to get the particular phenotype I care about? And as I said, before, these models come with provable guarantees about what is that, if you have an attention layer in there, this attention layer can actually be interpreted causally. So I can really go and learn these gene regulatory networks based out of what is used in there or what has been learned using attention. So those are the types of models we're now, of course, applying to all kinds of different applications, be it in rejuvenation, being in all kinds of cell state engineering applications. So with that, I will end. So this would, of course, not be possible without large-scale collaborations, in particular very close collaborations with cell biologists like G.V. Shivashankar, who we've collaborated with for over 10 years very closely. Then others at the Broad Institute, so that's, for example, cardiologists who work on that heart application. Then people on the more statistical side, like Matthias Durton. And then people at IBM Research, where we did a lot of work together on causal representation learning. Munzer Dale, so people who do more things about control, right? Because we're trying to control the system to move to a particular state. And hopefully, I'm able to convince you through this that it requires many of us to get into to solving these important biomedical questions. And in particular, that the biomedical sciences are, not only uniquely suited to being one of the greatest beneficiaries of AI, but really also one of the greatest sources of inspiration for it. Okay, because the biomedical sciences do require us to solve new problems in machine learning and AI and data science. And we've for too long now been motivated by questions like recommender systems, say self-driving cars, online advertising, which might just have led us to other types of questions than the biomedical sciences really needs. And we cannot just simply blindly apply these models. In particular, for example, here we saw how much we actually need models that can really reason about the underlying system from a causal perspective. And thanks to amazing students in the lab who really did all of this work, and then, of course, funding. And thank you all for your attention, and I would love to take some questions. And so I think there are microphones somewhere. I think I see them. Yeah, there is there. Perfect. Please.
Hi, this is Juan Liu from Arizona State University.
Very nice to meet you.
Yeah, nice meeting you. And I have a question. You have this causal feature discovery. So you also mentioned that causal discovery is very, very difficult.
Causal structure discovery. Yes.
Yeah, so would this causal feature discovery lead to causal structure discovery?
So causal structure discovery is very difficult because we would have to do it on 20,000 nodes. So what I showed here is first you want to find a lower dimensional representation where you can do causal structure discovery. That's why, but it's harder in the sense that we first need to figure out what are the latent variables. But then at least we're trying to do it on a much smaller system. So as I mentioned that it will only work if the representation, if there is a true underlying lower dimensional representation, because that's the only place where we can do causal structure discovery. Thank you, that's a very important clarifying question.
Hi, my name is Farheen Umar. Thank you for the great talk. I was wondering, when you're training these models, so for example, you said that you're looking at skin cells, but then you want to extend this to other type of cells. So when you're training these models, how important is it to have representation of all different types of contexts? So I assume that if you only train on skin cells, maybe you want to also use some bone cells or some other types of cells, because how would we discover latent features from different modalities then?
Yes, very, very good question. So the thing is, even within skin cells, there is a huge amount of variability of skin cells. But it is very, very important that we understand the underlying space, that maybe bone cells are, in fact, very, very different. And so in order to generalize to these, you will do a very poor job. And so having the right representation of different cell types is another important question of how do I select these so that they can then really span afterwards the whole variability of different cell types. That's similar to other generalization questions where, the data sets that we're putting in are really, really important. Yeah, but I think it goes even beyond because we want to generalize to new perturbations or interventions as well.
Hi. Yes. Given that design of experiments is so pervasive, especially in the physics domain, as well as even e-commerce or other non-biological domains. So going back to one of your slides where you showed the architecture, I'd like to know how can one, not from the biological field, apply the same theory? So for example, would it be as, say, simple as switching out the encoders and decoders? Because I'm very much inspired by that and would really want to apply it to e-commerce. Thanks.
Yes. And I think that is a very nice application where you actually also care about causality, right? You care about predicting the effect of an unseen intervention. And so I showed you two examples also for heart MRI and heart ECG, just to hope that others, to show a little bit more that these methods can actually be generalized to other settings. And in particular, heart MRI and ECG was about exchanging the encoders, decoders, because now it's a movie, right? An MRI is a movie, and here we have a time series. And so, yes, I'm hoping that people would use these types of models also much more generally. But you would need to have a good representation of your perturbations as well. For us, that's genes or chemicals have a good representation of a molecular structure. So that will be the question in the other types of applications.
I have a question here. Yes. So actually, I have two questions. So one is more on the technical side. I think I'm slightly confused because one thing you said was that if causal relationships are those that appear in multiple modalities, but then on your slide when you're showing the theorems, you also said that there are things that are specific, like causal relations that are specific to the modalities, and then those that are shared.
Yes.
So can you speak a little bit more about that? So that's one question, and the other question is that is there any hope of getting more insights at a gene level, or this is more just prediction from interventions?
Yeah, really great questions. So the one about, first we did the simpler problem. We thought that's all we can do. Just only looking for whatever is invariant to all of the different modalities. Okay, so that was one shared latent space. But then our theorem said, hey, we can actually identify more. We can even identify latent causal variables that are modality specific. Then we developed algorithms that can actually now disentangle. Now we have some causal relations that are specific to a particular modality and some that are shared, and we can actually disentangle them. So that's where I think the theory was so interesting that it then led us to actually a whole theory on, hey, I can identify what is specific to one modality. I can use that for experimental design. Now I know, for these downstream tasks, I actually have to measure this particular variable. And the other question, sorry, I don't remember. But maybe I'll take another one here.
Yeah,
Oh. Should I? Let's do it afterwards, so then maybe we do one more here.
Hi, Tanya Berger-Wolf, Ohio State University. Great talk. I have I have too many questions, but I'll start with the one. You know, the key word in studying biological systems is systems.
Yes.
And so studying any part of it in isolation still gives good insight, but still, you know, it changes when you go to the systems level. How can you go from studying and understanding causality in individual parts, in individual contexts, and individual processes to kind of the composition of that to the systems level?
Yes. So I think what I'm hoping to also get through here is actually that having many different cell types in already when you're trying to learn the model is important because of, say, for example, this non-Gaussianity assumption. So it's actually easier because I'm looking for things that are invariant. It's easier if you're adding in all of the different systems that you're measuring. Now, there is another question of like systems being cell level, then tissue level, organ level, et cetera, which is maybe the question that you're referring to. So in fact, we have, you know, similar models. I didn't put the references there, but you can actually do this also by taking into account now you have, say, a graph neural network on cells and how they're neighbors to each other, which you can now also add in here. So building the system up and having everything together in one representation is actually, again, very helpful. You're looking for invariances across different scales there as well. But very important problems. I'll take one more until, oh, I should stop. Okay, I'm told to stop, so I will stop here. Please come find me after the talk. I'm happy to talk to everyone.